Spermatogenesis is the formation of a sperm, and this is a highly specialized cell. It has mostly nuclear material. The remnants of the centrioles, the spindle that pulled half of the nuclear material apart, okay, giving it half the number of chromosomes. And some of the spindle still remains, which is now covered by the membrane, which is going to form the tail. And the way this works is it spirals. It spirals. So because it's spiraling, it's surrounded by a mitochondrial spiral to keep giving these proteins the ability to change shape via the energy. The sperm has what's called an acrosome covering the head. And the acrosome is filled with enzymes that are going to allow the sperm not only to penetrate the egg, but the egg has a layer of follicular cells called the corona radiata surrounding it. It's going to take hundreds of sperm to start breaking this apart so that one sperm can get through this clear zone called the zona pellucida to the ovulated oocyte and then penetrate the oocyte. Once the sperm penetrates the oocyte, no other sperm can do that. The membrane of the oocyte changes and is no longer susceptible to the enzymes. Now, spermatogenesis occurs in the walls of the seminiferous tubules. It starts on the outer portion and goes towards the inner portion. It starts with undifferentiated cells called spermatogonium. And spermatogonium will undergo mitosis, meaning the making of another spermatogonium, and then one will stay behind and one will differentiate. That way you have one that can do that again and again and again and again. Quite a bit different from the female. And this is done because these cells here, called the interstitial cells or the interstitial cells of Leydig, produce testosterone. And testosterone is what causes this differentiation process. Now, as you can see, there are blood vessels in this layer here, and if it were not for the sustenticular cells of Serotoli, a lot of the testosterone would end up in the blood vessels and go elsewhere in the body. But the sustenticular cells of Serotoli secrete ABH, androgen binding hormone, which keeps the testosterone in this area. The sustenticular cells do something else that is equally as important. This looks like no other body cell whatsoever. This looks like a foreign invader. This looks like a parasite. It has a tail like a parasite. It has different nuclear material. It has different enzymes. It has very little organelles other than mitochondria. And so our white blood cells would attack these if they were not prevented from doing so by the sustenticular cells. They form what's called a blood-testing barrier. So the differentiation starts taking place, okay? And finally, in the lumen have a sperm called a spermatid that has completed spermiogenesis. Now, this is not motile. This is not motile, okay? And in fact, to get from the seminephros tubule to the afferent ductule to the reed testes to the efferent ductule to the epididymis requires that these tubules are lined with cilia. And the cilia move the sperm to the epididymis. The epididymis, okay, secretes a fluid that keeps the sperm from doing what's called capacitation. Capacitation means becoming motile. So they keep it in an unmotile because they want to keep it there, nourish it, and wait for when it's going to be ejaculated. If it isn't ejaculated, then they will degrade it and recycle it. And this epididymis, okay, head, body, tail, okay, head, body, tail is 27 feet long. It's a coiled tubule that's 27 feet long before it even gets to the vas deferens. Okay? So that's where the sperm are stored. That's where they mature even more. They do not become completely capacitated until two things happen. They mix with the fluid of the seminal vesicle, and they are deposited in the female vagina. And secretions from the vagina combined with secretions of the seminal vesicle, complete capacitation, which means now it can start expending energy to move. So it's kind of a conservation process. Do not expend any energy going through a 27-foot long tube, and then maybe another 18 inches for the seminal vesicle, for the vas deferens and the male urethra. It's not